First session, which is about the biology of organisms of the Eastern Tropical Pacific. And our first speaker will be Kathy Teles from UC San Diego. And it's about zooplankton displacement and bioluminescence. Thank you, Kathy. Let's go. Bill.
Okay, hi everyone, my name is Kathy Teles, and today I'll be presenting zooplankton's vertical migration and bioluminescence activity in Playa Picote. To give you guys a nice introduction about zooplankton, it's important to the marine community as they forage for plankton, algae, and bacteria. They're also incorporated in nutrient recycling as they produce nitrogen and phosphorus for phytoplankton, and they alter fish biomass through prey and predator interactions. In terms of vertical migration, it is one of the largest migrations in the world from the deep sea level all the way to the surface level. Zooplankton have this behavior as they are trying to avoid predators. So what they do is during the daytime, they migrate to the bottom of the sea level in order to seek refuge in deep water, and then they'll vertically migrate to the surface in order to feed. For bioluminescence, it is an behavior in many marine organisms. Its importance, again, is for nutrient recycling, biodiversity, and predator-prey interactions. And again, zooplankton are species that have this behavior. So then, because there was a lack of research in the Northwest Pacific Ocean, it led me to my research question: what is the relationship between day versus night marine zooplankton vertical migration and observed bioluminescent flash frequency? In order to conduct my methodology, I started first at knowing my site. So the red pin is Playa Picote on the Northwest Pacific Ocean of Costa Rica. Midshore with the white pin is where I did my data collection. And then in terms of my materials and methods, I had snorkeling gear in order to go into the midshore, a plankton net attached to a 40 milliliter tube to collect my water samples. I would start with bottom level, which was 4 meters below the surface, and then I did surface level. Water samples were then put into an observation tank with ocean water that was filtered with a mesh water filter. The reason for the mesh water filter was to ensure that there was no other zooplankton or organisms in the observation tank, to ensure that the 40 milliliter water sample that I pushed into the water tank was just the flashes in the observation tank. And then the other 40 milliliters was in the dissecting scope to see the species abundance and the morphospecies identity. And then everything was put into Excel and statistically analyzed using SPSS version 31. In terms of my results, initially looking at the graph, you see the mean species abundance and then the water column levels as well as the time of day. Surface, nighttime had the most mean species abundance. However, a two-way ANOVA showed that there was no effect on time of day on species abundance, nor depth on species abundance, nor was there an interaction between species abundance and time and depth. And then in terms of mean flash frequency, there is bottom and surface as well, day and night, where bottom at night had the most flash frequencies. It's important to note that there are no daytime flash frequencies, so then running that two-way ANOVA showed that there is an effect of time of day on flash frequencies. However, there was no effect of depth level on flash frequencies, as well as no interaction between time of day and depth. Next, I have flash frequencies and species abundance. This shows pretty much Spearman's row correlation showed that there was no relation between flash frequencies and abundance. However, you can see that there are certain species that bioluminesce, and then there are some that don't, which is an important thing to know. And then in terms of my daily migration patterns, I have a number of species with the water column levels as well as day and night, showing that there are some species that vertically migrate, reverse vertically migrate, and there are some species that stay in the same position, as well as the abundance is different within the water column at different times of day. So as previously mentioned, there was no effect on depth in average morphospecies abundance. So another study saw that there was also no effect until they started considering life stage. This was something that was not considered in my sample, so I think further along should be considered for further research, as there were certain organisms at different life stages, such as like crabzoe or fish larvae. Daytime had an effect on flash frequency, where depth did not, so this could also mean that, for example, there were no daytime flash frequencies, so abiotic factors such as water temperature or daylight might affect bioluminescent behaviors, which was also something not considered in this research that could be used for further research. There was also no correlation between flash frequency and total zooplankton abundance. This is very important to know because, as previously shown in the graph, there are certain morphospecies that do bioluminate and some that don't. So there really shouldn't be a correlation between the two. And then the differences in zooplankton positions reflected vertical migration, as well as reverse vertical migration and no change in position. Something that this research also didn't note is the fact that there are specific predators that could be at different levels of the water that can affect the zooplankton's position, so that could be looked into for further research, as well as water temperature and other factors. I'd like to acknowledge my primary advisor, Leonardo Chaclamon, for his support and for helping with data collection. Naomi for being my secondary advisor, helping me with any revisions. Frank for his support throughout the program. The Diving Center, Guatemala, for their help. And then my host family, Familia Vasquez Blanco, for their support. Here are my references. Thank you. And then I also have some of the morpho species here that I observed, just to show you guys some of the diversity that was seen under the microscope. First, we have the Cirrolana cranchi. It looks really random. It looks almost like lice or something in the water. The crabzoe, which is pretty much these two are pretty much the lower developmental stages that I was talking about, the crabzoe and the fish larvae, that was not considered into the results. However, it is very present. And then this is the bryozoan. And yeah, thank you.
We have plenty of time for questions. Any questions?
When you were observing flash frequency during the daytime, did you look at the tank in a dark room so that you could see it?
Yeah, so the tank was either in a dark room in Casa Verde or in a dark room in the Playa Cote area. It's important for us to have kind of more controlled variables. So it was a 15-minute interval in a dark room, as well as something that I forgot to mention was there was this. I also saw that there was this factor of disturbance where I kind of, if I ever shook the bottle or I shook the tank, there was more flash frequencies, which is also something that further research can look into if disturbance is a greater factor in bioluminescent activity.
Interesting, thank you.
Any other questions?
Was there any species that like surprised you within the tank?
I think the crabzoe was the one because Leo and I looked at it at first and we were like, maybe it's related to like a worm or some sort of predator, and then it was really hard to figure out what exactly it was because it is in such a development, like very low developmental stage. Like, this isn't even what a crabzoe and like books show, but you can see the facial features and then also the fact that it has almost like spiny features in them. And that's also another thing. Taxonomic bias was something that should be considered in this research. Obviously, my bias would go into trying to figure out what morphospecies I identified, so that's also very important.
So you mentioned that this is one of the largest migrations in the world. Can you just quickly review for us again what the adaptive significance of the migrations are?
So Dow vertical migration is a synchronized migration that many species have. So pretty much in terms of zooplankton specifically, their predators are able to locate their movement as well as some of them are eco-sounders. But there's a lot of fish that need ambient levels of light in order to predate. So what zooplankton does is take advantage of that factor. So during the day where there's ambient levels of light, it will seek refuge in deeper waters that are darker and kind of hide in those deeper waters. And then they obviously need to feed, and that their foraging is usually done in the surface area because that's where their food is more abundant. Therefore, during the night where they see it by less ambient levels of light, they will migrate to the surface of the water and feed at that time. And then the cycle continues.
Great. Thank you, Kathy. Yes.
Thanks.
And our speaker will be around after the presentation. And now is the time for Lana Lilienstein, and she is talking about the bioreceptivity of coastal coralline algae and the potential enhancement of coral restoration. And she is from UC Berkeley. So, welcome, Lana. Right? Thank you so much.
Hi all. As most of you know me, my name is Lana Lilienstein. I want to apologize at the beginning. There's a little bit when transferring it from a slides presentation to a PDF. Some of the formatting changed a little bit. Nothing that will prevent you from reading it, but it's a little bit off. My topic today is going to be on crystallose coralline algae, otherwise known as CCA. And I'm evaluating the substrate bioreceptivity for its recruitment. So, first of all, what is CCA? Why does it matter? Why did I choose to look at it? I wanted to look at a big global issue that's going on that affects people all over the world in terms of economic factors. And I chose to look at how corals are bleaching and kind of their ecosystems are declining. And so thinking about restoration factors, how are we improving these systems and enhancing them. So CCA is actually found to increase coral larval sentiment through biochemical signatures. And this is an old version of the slides. Sorry guys. Yeah, and then by hosting beneficial microbial community across its list. So this kind of led me to my question: what naturally occurring substrates best encourage the development of CCA algae? So in order to look into this, my methods were to kind of evaluate 3 naturally occurring substrates that are pretty prevalent in the landscape. So 1, broken coral fragments, 2, submerged marine rocks, and then 3, live hexaplex shells. They were all ranked on a scoring system from 1 to 5 to kind of measure how much coverage of CCA there is prevalent on these substrates already, and then given a ranking. Shells and coral fragments were all different sizes, so I didn't use the same quadrant method that I did on the rocks, which was a 10 by 10 inch. Instead, it was more just a ranking system, so I could get a more accurate grade. These were some of the substrates. So on the top, you can see one of the hexaplex shells. You can see the quadrat measured on the rock, some of the coral fragments, and So my result for this part of the project was coral fragments that I found they were in low sediment areas, and they often scored around a 4 to 5, 25 out of 35 of the measurements that I took were 4 to 5, and then they averaged an 80. Hexaplex shells in low sediment areas where it was more rocky averaged around 72% of coverage, and then this kind of changed as soon as we went into the higher sediment zones where all of the live I couldn't do the same number of them, which is why there's a smaller amount of those in the low sediment, the high sediment area, because they're not as concentrated. But the amount of CCA coverage dropped significantly when the sediment was there. And then rocks, which were all raised away from the sediment, were kind of all over the place, but were around 50% of coverage. I also did a second experiment, which was deploying settlement traps into the water. So I basically created these experimental trays so they all could have the same amount of surface area. And there were 2 sets of triads, one of which was coral fragments that had been previously beached and they were all cleaned. Same with the hexaplex shells and then same with the rocks. And they were left for about a week to see if anything would colonize. And once they were brought out of the water, the brown and green algae that had accumulated was then wiped off to the CCA. What I found after removing them was the ring rocks had no CCA development. The hexaplex shells had a very small amount, around 2 millimeters in total, both in isolated patches, and then the coral fragments had around 8 millimeters of aggregate growth, again, all over different pieces. What was interesting about them is whenever the CCA would develop, it was inside the coral pores, kind of like a microorganism. So when I was taking all my measurements, I was trying to stay more quantitative, but there were also a lot of qualitative things that I saw. So beyond just substrate type, sedimentation has a huge effect, like how I was talking about earlier with the development of CCA. As soon as sediment is brought back into the equation, the CCA diminishes largely. And then herbivore presence also has an impact. So in rocky areas when the larger marine rocks had sort of holes where urchins could live in, or smaller fish, it would often be cleaner and you could see a much bigger presence of that CCA algae. And then light availability also had an impact, so that other algae species that would colonate on top, since CCA is an encrusting species, they could grow on top of it. As soon as the light was taken away, those algae would disappear, and the CCA was pretty prevalent. So the certain structures that had the most colonization tended to be ones with the calcium carbonate skeletons. So something that I would want to look into in the future and see in other research is it specifically the calcium carbonate that is like funding the CCA growth, because CCA is primarily also composed of calcium carbonate. And if so, how can we create this into a paint that maybe we could swab on structures before we attach coral fragments, so they would hopefully encourage those fragments, like when those fragments are more developed, to then attract the larvae. And then also what we talked about earlier, the role of topography and texture. How can we kind of create that homes for CCA so they can settle, like areas where they want to settle in within structures, and then how can that things similar to like those coral pores be replicated on bigger, larger designs. And then also one of the biggest factors that we saw, which the sediment. How are we raising these structures and making sure the sediment isn't necessarily smothering them and smothering the corals on these traps? Yeah. I wanted to thank the UCAP staff and everyone in total. I'm not from a biology background at all. I'm from architecture and design. So this this was a new process for me. And everyone was very helpful and just helping to explain what different ways to kind of approach this in an experimental sense using quadrats. I wasn't familiar with that before, or just lots of different things. And also the Laura family. My host mom was very kind throughout the process, and she gave me a love for Guanyu and Keel, and just having her there and just understanding why it is so important to protect these communities through protecting these areas that fund them is so valuable. And then also the Laura brothers for helping with the vote and actually getting us out there.
Excellent. We have 3 minutes for questions.
Since you mentioned these live shells, I was wondering how common it is in nature for these algae to grow on live mollusk shells and what impact, if any, there is on the mollusk.
Yeah, I'm not exactly sure if it has an impact on the mollusk. What was something that was really interesting was depending on the mollusk species, there was a different, or at least I can't say for sure because I didn't take measurements, but visually there was a big difference between CCA coverage and mollusk species. There was a specific more round species that had like almost like a barrier on the outside of the shell, which seemed to prevent a lot of the CCA from growing. And then the hexagon shells, just it would grow all over.
Another question?
Especially interesting to know that in the substrates that you put out, the CCA was colonizing, especially the areas with cool pores. And I'm wondering if you have any insight as to why that is. Apparently they don't have flagella, they don't have cilia, so they're completely dependent upon micro water currents to end up where they do. And it also relates, I think, to one of your interests of perhaps, well, you said it's about painting surfaces, but setting things up so that the water currents get them to a solid surface that's suitable for colonization. I wonder if you have any thoughts about that.
There was this really cool paper that I read before even going into CCA about coral and about how currents affect the larva actually settling and the movement in which the water goes, if it's right to left or if it's kind of moving all over, if there's no water movement, impacts the settlement of the larva. So I wonder if CCA almost has a similar mechanism where the direction of water movement and its velocity and stuff has an impact on its development. So if the water isn't as easily like pushing against it, it might have an impact on its like growth pattern.
I also have a question. In terms of your settlement traps, do you think that the size of this could also influence the amount and the cues that the algae are using for finding a place?
I think I'm honestly not completely sure. I haven't delved into the research enough to answer your question fully. I'm not sure. I would want to see kind of the contrast between having a larger amount versus a smaller amount.
So,
See, guys, look. You're like, ah. Oh my god. Someone said I'm red for the Look at this dude. Who is that?
So I apologize for the inconvenience, and our next speaker is Anna Kessler. She is from UC Los Angeles and is going to talk us about the marine hermit crab's homing behavior.
Hi, my name's Anna. I'll be talking about homing behavior of intertidal marine hermit crabs. To start us off, the intertidal zone is the area between the shore or the coast and more of the land. This alternates between marine and terrestrial conditions as tide rises and falls. During high tide, the land is submerged in water, and then during low tide, it's exposed to environmental conditions, putting it in terrestrial pressures. Tide pools provide refuges from desiccation and heat during low tide. As organisms experience rapid changes in temperature, salinity, moisture, and predation pressure during low tide, and understanding how these animals locate and return to refuges is imperative to understanding their survival in the intertidal zone. Marine hermit crabs in this study were Clibinarius albidictus, which occupies the upper intertidal zone, Calcinus obscurus occupying the mid or lower zones, and both these species use tide pools as refuges during low tides. However, it's unknown if the species exhibit homing behavior. Homing behavior is defined as the process by which animals return to central places such as nests, territories, refuges, after foraging or other movements. This brought me to the question, do intertidal marine hermit crabs exhibit homing behavior, and what factors influence this? To investigate this, I went to the tide pools in Islita and Carmen Keel on the Pacific coast of Costa Rica. I selected 3 pools on varying sides of the inlet, and then every day I marked crab returns, which I quantified by marking each crab with a corresponding paint for each pool, and then individual markings for individualistic recognition. I also took note of pool depth every day of collection, salinity, temperature of the pool, as well as temperature on the land. And each crab I moved randomly 4 meters, 3 meters, and 2 meters from the pool in the same direction in each collection to measure whether displacement had an effect, as well as collecting data on their displacement from the pools after their day of data collection. And then I quantified return to the original pool as a homing success. Overall, there were 44 out of 98 individuals from my collection that returned to the pools. However, the treatments of displacement had no effect on these return rates. And the individual probability of return decreased as days increased from their moment of collection. Their ability to travel short distances, or it suggests that they have an ability to travel short distances, and that the 2 to 4 meter treatments also within their normal range of movement, considering the fact that displacement had no effect on their ability to return. And crabs are often absent from the pools during high tide and return during low tide, suggesting foraging movement, which would also affect their ability to have
factors rather than just displacement and origin of the pool. These environmental pressures that I investigate are pool depth, salinity, pool temperature, and outside temperature. Pool depth had no effect on return rates. However, deeper pools do tend to be cooler, so there is still a chance that they have an effect on these crowds with return rates. None of the pools that I investigated had a large range of depth, the range from 3 centimeters, 6 centimeters, and 13 centimeters. However, in one of my pools, there was a neighboring gulch, you can kind of see it here, that connected to the ocean, allowing possible advantages to these crabs for them to prefer to relocate to this gulch rather than to return to the original pool. Also, there could be a sort of connectedness between these pools that crabs just roam between them interchangeably, recognizing both of these places as sites of common behavior and use them as different, or as for different purposes. The gulch may provide better foraging, better temperature regulation when it's hot out, and just better connectedness. Salinity also had no effect on return rates. Salinity remained relatively constant throughout my study, and it fell within the tolerant levels of the species. But if this level exceeded or left this range, it would probably pose a risk to their return rates. And then also, hermit crabs have water retention in their shells that minimize the effects of fluctuations in salinity. So this may not even pose that much of a threat to influencing their own behavior. Pool temperature did have a weak correlation with return rate, whereas temperature of the pool increased, return rates decreased. And the same goes for outside temperature, whereas outside temperature increased, return rates decreased. And then I plotted, or this is how temperature changed over collection time during the days of my study. And there were times, as you can notice, where pool temperature actually exceeds land temperature. And it was very often in these moments that crabs were seen outside of the pool rather than in the pool. They were often seen in crevices, or in such as one of my pools I mentioned before, in the deeper gulch. Their leaving the pool during these high temperatures is likely due to their water tolerance. I found in past studies that these crabs have a tolerance around 33 degrees Celsius for leaving pools when the water exceeds this. This is due to water as a better heat conductor than air, so it puts them at more risk of water retention loss and desiccation risk if they remain in these hotter pools. So they can go and shelter in shade or possibly deeper gulches or go into the ocean, which will affect their homing behavior return, as it's not advantageous for survival, favoring if they stay in pools. Overall, this suggests that homing behavior is supported to an extent, with 45% of the population returning to their pools. This moderate return rate suggests that hermit crabs may use spatial memory or orientation mechanisms to relocate to familiar habitats, due to displacement having no effect on their return rates. And their ability to return after high tide to these pools. Temperature did show the strongest trend among environmental variable measures, although none of them were statistically significant. This relationship is important. And it suggests that there's a preference for suitable habitat. I saw many crabs dig into the pool and leave the pool when it was too hot. This could be further studied to understand this behavior and how they assess quality of pools. And then homing behavior appears to be flexible and likely influenced by multiple factors. For example, I saw crabs return one day and then the next day they were gone, and then they come back the next day. So there has to be some sort of assessment and understanding if these pools are safe for them to return to or if they should go somewhere else. As I said, future directions, they can expand displacement distances to understand the range their cognitive map holds, as well as testing for a threshold exit for pool temperature. Understanding how these crabs can tolerate heat fluctuations is very important in the face of climate change and how heat may affect these species. Also, as I said before, studying hermit crab pool assessment and understanding how they assess pools and what quality of shelter they will move to next if their pool they deem is not adequate based on environmental issues. I'd like to have to thank you to my advisors Leo and Frank and to the Castro Campus family for welcoming me into their family. Thank you.
We have 2 minutes for questions.
For the number set that the crabs would choose to inhabit and the pools were too warm, were they deeper enough? Like, what were the actual?
I have a picture of one of that. Right here, it was kind of like a nook. It was probably like this deep, and it wasn't necessarily like deeper in the ground. It was offered a lot of shade, and I think there was like moisture retention that helped keep it cool. Like right here, protected like from evaporation.
Was there some nooks that they preferred over other nooks, or were they all scattered pretty evenly?
I'd say this nook was like kind of wide, but they all favored like this one like deeper section. Like routinely, they would go on the same page with the nook. That'd be cool to investigate like I said, like quality necessity of like refuge.
In terms of you labeling the shells, how effective was your labeling method? And
There were like times where I noticed that the nail polish did come off. We would find crabs that had like one bad nail polish from the previous day and they lost it over time. So there is like that air that was probably likely that many crabs had selected from my population where they lost their markings, so then they effectively would never observe again.
did you say how many colors in the crabs you have?
99.
99 crabs?
99 crabs.
And how many colors? Like what color?
Probably like 15.
Whoa. Okay.
You're still having time.
I forgot mine.
Okay, I also have a question. Is there, beyond the capacity of looking for more suitable places, is there any trait that can explain this flexibility regarding homing behavior that these crabs has?
You mean like what gives them their ability to recognize?
No, I mean like the ability of select or not to going back to a pool, thinking that they need to have water for being alive. Is there any trait that they show, like retention of moisture?
Yeah, they retain moisture in their shells.
Sorry, I was out to close the door and I may be missing.
You mentioned that sometimes there would be a crab that would go and then not come back the same day, but would come back the next day. Did you mark that as a success for homing behavior or a failure?
As a success.
Okay,
If they return to pool, that was a success.
Thank you so much. We don't have enough time for questions. This is not related to the computer.
Check my new fellow.
During the break, the Nacho will pass around this creature.
Sorry again for the technical inconvenience. Our next talk is intern for Selena Malot, and she is going to talk to us about the appearances changes in vestibular chromosome, the chromosome.
So many animals have the ability to rapidly change their color. This includes octopus, crabs, chameleons, and others. This color change is mediated by chromatophores, which is specialized pigment-containing cells that are under the control of the nervous system. The subject.
Okay. The subject of my study is the blue-spotted cornerfish, is also one of the animals able to exhibit this behavior. They are characterized by an elongated body with a long tubular snout, and they contain bright blue stripes and dorsal patterning. They're also able to exhibit a wide range of base coloration, ranging from a light tan to a darkish red brown. They're also able to exhibit this stripe patterning, which is a form of disruptive patterning or camouflage, in which the body of the animal is, the outline is disrupted and blends better. Previous EAP student in 2019 found that this striping behavior is only seen when the cornerfish is stationary. Further and more interestingly, they found that the color of the cornerfish is able to match their background substrate. So, what if this color change was more than just to passively match their background? Sepal color change was more extensively studied, and it is known that their color change is not only for camouflage, it's triggered by their ecological interactions, such as their fleeing from predators when they're hunting, mating, or communicating with conspecific individuals. So this led me to wonder, what if this was the case for the blue-spotted cornerfish? My central question of the study is whether the appearance of the blue-spotted cornerfish is influenced by their ecological interactions, including fleeing, hunting, and communication with their conspecifics. To analyze this, I did my study in 2 locations, in Playa Jicote and Bajo Rojo in Cajimiceo, Costa Rica. For my method, I studied for 10 days, and during those 10 days I conducted many trials. Each trial consisted of filming the cornerfish for 5 minutes or more with a GoPro 11 on a GoPro stick. After 5 minutes, I would dive down and pursue the cornerfish, simulating a predator. For trial, I also recorded the following data: the date, time, location, the size of the cornerfish, and whether they were alone or in a group of others. For the first part, I conducted a coloration analysis. This included a brightness analysis of both the cornerfish and their background. This was done using image J, and for the cornerfish, I was able to carve out the entire body of the cornerfish to get the average brightness. To calculate the background average brightness, I made a rectangle that you can see here in black around behind the eyes and copied it into 6 locations right around surrounding the background surrounding the cornerfish. Image J gives a value of average brightness from 0 to 255, where 0 is black, no light pixels, and 255 is bright, only light pixels are present. I also conducted a coloration for the color analysis, both on the base color of the cornerfish and also their blue stripes and dot patterning. This was done using Photopea eyedropper tool which is able to give an RGB value as seen in that picture there. RGB stands for red, green, and blue, and their values again range from 0 to 255, where 0 is that there's no pigment of that color, and 255 means that all the pigment is present in that color. So black would be all zeros, and then white would be all 255s. Okay, so for my results. First, I wanted to make sure that the cornerfish actually do match their background subject. This was observed by the previous AP student, but not really quantified. So this graph here on the x-axis shows the background brightness, and on the y-axis the cornerfish brightness. As you can see here, there's a positive linear line showing that as the background brightness increases, so does the cornerfish brightness. This indicates that there is an impact on the brightness of the background to the cornerfish brightness. Now, to answer the central question of this paper, does cornerfish base color change after being chased? And the answer here is yes, it does. If you see here on the x-axis, there's individual cornerfish, of which there are 18, and on the y-axis is the difference in color value. What this means is the RGB values before minus the RGB values after. So a y-axis of 0 would mean that there was no color change, and a negative values, as seen in a lot of these ones here, shows that the cuttlefish after being chased is brighter. This was also the case for the blue dorsal stripes and dots. As you can see here, there's a lot of variation, none of them are really at the 0, and 72% of the trials show that they actually brightened after being chased. Another interesting observation about these stripes and dots is that during hunting, the stripes was actually turned off. That's seen in the top image. And then after, like a second later, the stripes and dots come back. So this shows that there is color change after being chased. A majority of the individuals actually turn brighter. This is possibly due to a phenomenon seen in cephalopods called blanching, which is in response to stress or fleeing from predators, they become lighter. This also was seen in the blue stripes and dots. The brightness increased after fleeing. And it was also interesting that they were able to turn this patterning off while hunting. This is interesting not only because it wasn't known in any of the research I found that they were able to turn the patterning off, but it also brings up this idea of why the blue coloring is turned off while hunting and then increasing after. A possible explanation is that the blue light is most visible in the water. It is able to travel the furthest. So maybe the coloring can be easily recognized by the prey, which is why it's turned off while hunting. But when they're being chased, they want to increase the brightness. And the reason why is because the Lorentz hypothesis states that specific blue patterning allows for visibility or specific recognition. And this means that they were able to, when they were being chased, sometimes they would meet up with more members of their own species, probably as a way of protection mechanism, so increasing the brightness allows that to happen. Okay, secondly, I decided to look more into the striping behavior. For each trial, I looked to whether they exhibited the pattern. If so, was the cornerfish stationary when it happened? With what background substrate was the striping observed? How long they were striping, and then in comparison, how long were they stationary? So this graph here shows the different substrates and whether striping was observed or not. And across all these different substrates, there was striping seen. The only one where striping was not seen was in the sandstone and algae rock background substrate. Since previous EAP students found that the cornerfish only stripe when they're stationary, this was indeed also what I found. So what I was curious was whether if they meant that they were stationary, they would stripe more. But this turned out to not be the case. So this shows on the x-axis the total time spent stationary, on the y-axis the total time spent striping, and there was no clear linear correlation, showing that yes, striping is only possible when stationary, but just because they're stationary does not mean they're going to stripe. I also noticed different levels of intensity between the banding of the stripe pattern in the same substrate in the same cornerfish. So this shows their capability of changing the contrast within the band. So stripe patterns, as I mentioned earlier, is able to see in many substrates, even ones that are low contrast like sand. There are different levels of, this is against the notion that the disruptive patterning can only occur under the heterogeneous substrates. There are also different levels of contrast within the stripes, regardless of substrate, and this is unlike the cuttlefish, which is able to match their levels of contrast with the level of substrate. But this is not seen in my experiment. The last cool thing that I saw was that the cornetfish was swimming with a group of lisas, which is a mullet species. Previous research has shown that the cornetfish can hide behind singular herbivorous fish for their hunting. But doing it in the school like as such may aid in further camouflage and increase their prey and hunting success. Okay. So I would like to extend my sincerest thanks to the following people listed here, and also to my peers. These are my references. Thank you so much.
Thank you, Elena. We have 2 minutes for questions. Go ahead.
So, in these changes that you studied where the cornetfish changes color while being chased or while hunting, are those changes in the direction of looking more like their substrate or less like their substrate?
For most of them, I looked at ones that where the substrate did not change to make sure that that wasn't a factor in the reason that they were changing color. But it is also possible that as they change, as they're moving away, they match their background substrate. But usually what I would see is that they would maintain the same color as they sped away because they were focusing probably more on swinging than matching their background. But I did ensure for this part of the experiment that to negate that aspect, to only compare within the same substrate.
Some animals, like chameleons, will color change for temperature regulation. When you're doing your background research, did you see any studies that looked at that in cornfish or not really?
No, and in fact there wasn't really much about the color change behavior aside from the fact that they kind of match their backgrounds. In other species, or other similar species, like in the trumpetfish, they've shown that the mating can be influenced by their color change, but not much is really said in the cornfish.
Elena, do you know the app you're using? Which are the units of brightness or red, green, colors? So it's a value. But do you know about the units?
The units? I was also curious about this, but it's just the amount of pigment. So it's a universal scale from 0 to 255 for RGB, and it's based on the amount of pigment per.
My concern is that if those are linear things or they have a given behavior in the magnitude, because you are using them as a linear aspect, and it's something to collaborate. Thank you.
We don't have more time for questions. So our next speaker is Anya Christopherson, and she is going to talk us about the aggressive behavior of dancelfish through different intruders.
Hi, I'm Anya, and my study investigated the aggressive defense of algal gardens by dancelfish against intruders. So globally, we're seeing a decline in coral reef ecosystems due primarily to anthropogenic climate change. It is thus necessary to better understand the ecological processes and interactions that underlie coral reef ecosystem resilience. Damselfish are ephemeral herbivorous fish that have a really large impact on coral reef community structure, specifically in terms of algal biomass and the presence of other herbivores. These damselfish garden algae by defending a territory which they selectively weed out
species of unwanted algae and defending algal gardens from other herbivorous species such as sea urchins. The objective of my study was to examine differences in the aggressive defense behavior between the giant damselfish and the Acapulco damselfish against different intruders within algal gardens. So the test species I used were 3 equinoids or sea urchins, and then 2 species of herbivorous sea snail. So I looked at the giant damselfish and Acapulco damselfish in terms of differences in aggressive behavior. And the urchin intruders that I introduced were Diadema, Echinometra, and Lucidaris. And then for snails, I tested Hextoclex and Basurba. So my study took place off the coast of Guatemala at Playa Jicote in Guanacaste, Costa Rica. And to compare the defense behavior between the giant damselfish and the Acapulco damselfish, I first located algal gardens. I did so by observing the area in which individual damselfish was actively guarding and chasing away other fish from. And so you can see here in the first picture, the area of filamentous algae on the rock is the damselfish's algal garden. And then next, once I had identified a garden, I introduced an intruder, so either one sea urchin or one sea snail. And I then filmed the behavioral trials with GoPros for a total of 10 minutes per trial. And I later analyzed video footage looking at the latency to the first attack on the intruder, the attack duration, the bite rate, and then removal success. You can see it's very good. So a key result from this study was that intruder type influences damselfish aggression. Across the board, looking at the different metrics of aggression, the Acapulco damselfish appears to be most reactive to intruders, with the exception of the Diadema urchin. And this could be due to the morphology of Diadema. This species is a larger body size than the 2 other test urchins, and their long spines also possess toxins. And so it could be that the Diadema is just more physically defended. In terms of bite count, as seen here, we have on the x-axis urchin species and then predicted bite counts on the y. And in pink we have the Acapulco damselfish. And so you can see that for Echinometra and Lucidaris, the Acapulco does have higher bite counts, but in terms of the Diadema, the giant damselfish was biting more. And then similarly for removal time, we see that the Acapulco damselfish is like very quickly removing both Echinometra and Lucidaris urchins, and then those species of fish are taking longer in terms of Diadema. Removal success, we again have the Acapulco damselfish successfully in 100% of trials removing Echinometra and Lucidaris urchins, but in terms of Diadema, the Acapulco damselfish really struggles more with only like a 64% success rate. And then when looking at differences in snail introduced, we see that the 2 snail species and the Acapulco damselfish is attacking both species of snail more when compared against the giant damselfish. And so these differences indicate that the Acapulco damselfish may be the more aggressive species of fish. Another significant result was that bite count differs by urchin species. Again, this could be due to the morphological differences between the urchins. Since the Diadema urchin has longer spines, it may just be more difficult for the Acapulco damselfish to deal with. And then it could also be that Diadema is perceived as a greater threat by the giant damselfish, and as such is being attacked more. Diadema urchins have significant impact on
Additionally, differences in bite counts could be due to damselfish attack strategies. The giant damselfish was observed more to attack by biting the urchin and trying to break off spines, whereas the Acapulco damselfish was observed to bite and try to physically lift and remove the urchin from its garden. And so it could be that the physical defenses of the diadem urchins were just harder for the Acapulco to deal with. So urchin identity also significantly influenced removal time. Again, we can see here the diadem trials for both species of damselfish were much longer than the other trials. And interestingly, the UC-Doris urchins were removed the most quickly by the damselfish species. This could also be attributed to this urchin's morphology. The UC-Doris urchin, as seen here, has more sparsely dispersed blunt spines, and these characteristics made it easier for the fish to eventually just pick up and remove. All trials with the UC-Doris urchins resulted in the urchin being completely lifted off of the garden, as seen here. And then this finding also corroborates results from Irving and Rickman, who studied 2 different species of sea urchin in the Galapagos, and similarly found that UC-Doris urchins were most quickly attacked and removed by damselfish. So in terms of removal success rate differing by urchin species, I found that urchin species significantly influenced the success rate. Again, this can be attributed to diadem's morphology and just the species being larger and harder for both fish to deal with, as both fish have lower success rates moving diadem. And then in terms of damselfish attacks on snails, this finding, while not statistically significant, may also hint at greater aggression by the Acapulco damselfish. For both the hexaplex and the bacillus snails, they were attacked more so by the Acapulco damselfish than by the giant damselfish. And defense behavior may also be driven by intruder morphology. The hexaplex is a more three-dimensional intruder and has a more spiny appearance. So overall, I found that intruder types significantly influenced the probability that the intruder was attacked. This graph shows snail versus urchin, and then the predicted probability of attack estimated by a generalized linear model. And so this is basically putting together all urchins in all trials, and then all snails in all trials. And urchins were attacked 100% of the time in the trial, but snails were only attacked an estimated 43.6% of the time. So damselfish are ecosystem engineers that strongly influence the structure of coral reef communities and fish assemblages. Through the aggressive defense of their algal gardens, they can alter which herbivores have access to algae and other resources on their reef. And herbivore diversity is known to be important for maintaining reef health. So understanding better the interaction between damselfish and other intruders can provide us insight into the broader ecological processes occurring on reefs. These findings also contribute to our understanding of species interactions on coral reefs. With growing climate change, it's important to have a better understanding of these processes to help inform conservation management practices. I would like to say a huge thank you to Leo, my primary advisor, for all his help and guidance and all the time spent reading urchin spines out of my fingers. Also, thank you to Naomi, Frank, and Fede, and my host mom, Shirley, for so graciously letting me to home. And thank you to the Laras and the rest of my peers. Thank you.
1 minute and 15 seconds for questions. It's not a joke.
Are there areas of the reef that are not claimed as territory by damselfish, or where do these other herbivores survive?
Yeah, so you see in some of the photos. So yeah. So you can see here, like this is a good example of a defended territory where you see the filamentous algae growing, but then on the other side it's just barren like rock or rubble. And then you can see, it's kind of hard, but you can see an aggregation of diadema urchins. And so I noticed that I guess the shallower areas of the reef did tend to have more algal coverage overall and more damselfish. But areas that were not defended by the damselfish, like a lot of them had a pretty stark contrast between algae and then the female algae.
And second. Thank you. And thank you, Silvio. I just realized this when I see the methods from here, and this is what I was pointed out. Like, this shows this significant difference in the guarding of this organism.
Sorry.
Yeah. So thank you so much. Our last but not least speaker for this wet and salty session will be Kelly Knight, sorry. And she is going to talk us about the species composition and succession of fish in baits.
Hi, I'm Kelly. So yeah, I used a gated remote underwater video system with the acronym GRUB to survey fish communities at Bajo Rojo and Cote in the North Costa area. I did 12 trials across 3 different sites in 2 locations. It was shallow reef, mid reef, and sandy bottom. So coral reefs support high levels of biodiversity, and our areas with greater complexity tend to support higher species diversity and more resources and protection from predators. Understanding fish community responses to habitat differences is crucial because it provides insight into ecological importance of reef structure and it can inform conservation and monitoring efforts in tropical marine ecosystems.
So I utilize brubs as a non-invasive
Study,
method because you can
set the camera down and
leave it for a certain
amount of time.
which significantly decreases the fish's like wariness of divers and can allow us to see fish species that we wouldn't normally see if there was human presence. And what I wanted to predict was fish abundance in these areas. So I expected fish abundance to be highest in shallow reef habitats due to greater structural complexity, and higher fish abundance predicted at the Bajo Rojo site. Bajo Rojo was depicted as a rocky reef habitat with open ocean conditions, lots of surge and uplift. And Cote was a sheltered cove with a sandy bottom and rock coral reef structures. So this is why I expected higher fish abundance at Bajo Rojo, and I expected shallow reefs to contain the majority of fish species in abundance. So here are my 2 sites, and shallow reef, mid rock, and sandy bottom were measured across 1.8 to 5.6 meters. So this is the grub. It was made from a PVC pipe with a 1-liter container with drilled holes on one end, and it was weighted down to the bottom. I would leave the other end had a GoPro on it, zip ties so that all the fish that came without being there. So I would drop the device down and leave it for 30 minutes. And this is the amount that I would record for. However, I'd only have standardized observations for the first 0 to 5 minutes, 11 to 16, 21 to 26. And then when I open the container, I would observe the fish until all of the food was gone. Inside, I placed 2 chopped up mackerel, about 1-inch pieces in each, and the holes allowed for scent dispersal and for more predators to come to the bait. So this is the total number of fish observed at each site. Cote Shallow Reef had 86 individuals, Bajo Rojo Sandy Bottom had 78, and Bajo Rojo Mid Reef had 64. These are the highest abundances, and abundance patterns did not match my original predictions, which was for Bajo Rojo to have the highest abundance. However, both locations showed high abundance influenced by habitat type. The shallow reef habitat contained the highest structural complexity with large rocks, crevices, and refuge areas for fish. And the mid-reef area had more depth and complexity and consisted of mostly rocks, and the sandy bottom was mostly sand substrate with net 0 rocks. Here's a figure that represents that chart that I just showed. I also measured the species richness across habitats and sites. So there was a total of 30 fish species that I recorded, which represented 15 different families. The most species-rich families included Labridae of grasses and Muranidae of moray eels. This highlighted the importance of habitat complexity in supporting diverse fish assemblies. The wrasse were most diverse on tropical reefs because they fulfill a lot of different roles as cleaners, predators, and foragers. And moray eels enhance reef diversity and tend to inhabit crevices in rocky areas where they can hide. Cote had the highest richness in mid-reef and sandy bottom habitats, and Bajo Rojo peaked in richness within the shallow reef habitat. I also used, the species richness varied from 7 to 12 species depending on the habitat. And I used a Wilcoxon sign rank test to compare species between Bajo Rojo and Cote. The test statistic was 3 for t-value, and I used a 2-tailed p-value of 0.625. The p-value exceeds the significance threshold of 0.05, so the null hypothesis was not rejected. This means that there's no statistically significant difference in species richness between the 2 sites. So despite these variations, the fish communities were relatively similar in terms of richness across both locations. So these are some of the species that were unique to Bajo Rojo. We have the saber-tooth blenny, which was only found in the sandy bottom habitats of Bajo Rojo. They're unique species known for mimicking the rainbow wrasse, and they would often be seen following eels and taking off scales from them because they're aggressive mimicry. And the Panamanic green moray, which spends most of its time in cracks and crevices, which is really representative of the Bajo Rojo habitat. The jewel moray contributes to regulation of prey populations and also often found in these crevices. In the sandy bottom habitats, there were a lot of porcupine and pufferfish because they eat mostly crustaceans and worms and stuff found on the sand. And the white-spotted puffer at the bottom is especially notable because it
has a variety of trophic roles because it's an omnivore, so it eats algae, sponges, and small invertebrates. I also saw a long-tailed stingray at Bajo Rojo, which are known to just patrol the sandy bottom habitats. And I looked a little bit into succession of fish, but couldn't find too much significance other than chameleon wrasse, rainbow wrasse, and garlic serrano showing up first to mates. So this study found that habitat complexity drives diversity, and that reefs provide shelter and resources for lots of fish for predation, protection, etc. And browses is an important study or useful method for bait-attracted species. Some problems with this study is that the bait attraction bias would only attract carnivores and omnivores, and I didn't see very many herbivores. And I tried to find some habitats that had the same sort of features, but there were differences, obviously, and I had a small sample size. Future studies can include more trials, seasonal samples, additional habitats, and more herbivore-focused baits, and a behavioral analysis into the species. So overall, I found 30 species from 15 families. The richness ranged from 7 to 12 species. There was no significant difference between the locations, and the Brev successfully documented reef fishes, and habitat connectivity may support this diversity. Thank you to my UC EAP instructors and my advisors, and the Dive Center.
Any question?
You like eels? Yeah, they're pretty cool.
Any other question? What is a board?
A breath?
Sorry, yeah, bro.
Yeah, it stands for Baited Remote Underwater Video System. So it's just a bait and a camera on one. Thank you.
So this is the end of this first session, and now we have 10 minutes for a break. So we will see you again at 10:03. There is food in the back. Thank you for the help. Thank you for being here, and you are welcome.
Good job. Nice to meet you. We're not having a field show that lets you come in. Wait, can I while I'm back
So yeah, the flies lately are so I think you're
There's what? Guys. Guys, not my friend. I'm happy too. Frank said I can try, I just have to.
I'm not sure about the PDF.
Okay, we're going to start. Okay, now thank you for participating in the next section, which is the second about biology of terrestrial and aerial animals and their microbes. First speaker in this section is Ilse Vasquez from Riverside. She's going to speak about the distribution of visual wing damage across wing regions in the broad wing geometry demo. Welcome.
Hello everyone, my name is Elsa, as you mentioned. I will be presenting today the distribution of visible wing damage involved between geometry, moss, and I'm from University of California. Before I get into the damage, it is very important to know what type of moss I'm doing, and this is the family geometric moss. This is one of the largest moss families with over 32,000 species, and they are commonly known as inchworm moss. They're very known for having open wing position, and they are very important to ecological tropical areas, as they are very known for serving as prey for bats, birds, and many other predators, and they also are known for pollinators. And so why study these wing damage? As their wings are very essential for their flight, and as well as survival and their movement, and also just reproduction. And we are able to see what predators and how environmental stress can damage these wings. So this leads on to my question. Is visible wing damage distributed equally among the apical and medial basal wing regions of the broad opening geometric moss? And my hypothesis is that wing damage is not distributed equally across these wing surfaces because different wing regions experience different varieties of levels of environmental exposures as well as predation pressures. And my prediction is that the apical wing region would exhibit the highest level of visible damage, followed by the medial regions, while the basal regions exhibit the lowest amount of damage. For my study site, as many of you guys know, I did it in San Gerardo, Monteverde, Costa Rica, in the Bosque de Morelos field station. My sampling took days took during the days of May 4th through 8th, and I started around sundown, around 6 p.m., leading up to 11 p.m. or stuff. I was doing off and on observations for about an hour and a half each night. The moths were attracted using a large white sheet and 2 fluorescent lights. Throughout this time, these 4 days, I conducted 64 moths and I beat them. Adding on to my methods, each forward wing and hind wing was divided into 3 sections. The apical, medial, and basal regions. As you can see, the green is the basal, and then the yellow is the medial, and then the apical are the wing tips. And they were each divided into 4 scores of 0 being no visible damage, 1 being minor damage, 2 being moderate damage, and 3 being severe damage. These are some examples that were included in my studies, and seeing from the top as they are minor damage, the middle row being moderate damage, like a score of 2, and the last row being a score of 3 in the apical region a little bit. And these were all some examples of my moths. For my results, the hind wings, for the forward wings, I observed a clear pattern in both the left and right wings, and it showed that the apical regions had the highest amount of visible damage, while the medial had the moderate levels of damage, and the basal region had very little, or almost some of them were very undamaged. This suggests that the damage was more common near the outer portions of the forewings and the near wings of the base. And then for the hind wings, it showed very similar patterns to the forward wings, as both the left and right in the apical regions were the highest in frequency, and the damage was observed. And the mitral region showed intermediate levels of damage, and again, the basal showed a score of 0, indicating there was little to no visible damage. And my overall results, when all the wing types were collected, the apical regions consistently showed the highest levels of damage, while the basal regions consistently showed the lowest levels of damage. And then seeing in this graph, the red line showed the apical damage, and the highest was on the left hind wing, showing a 70.3% damage. That includes all of my moths. And overall, the lowest levels of damage were observed in the basal region. So creating a clear pattern where damage increased from the base region up to the tip of the wings. And then a chi-square test was shown that damage was not spread equally across the surface, and that differences among the wing regions were highly significant. And then as for my discussion, several factors may explain why the apical region exceeded the highest levels, and since the tips are located on the outer edges of the wings, they are more exposed to environmental conditions during flight and more likely to come into contact with predators as well as vegetation. And this could also increase the environmental aerodynamic stresses and may be more exposed to predator attacks. And this leads to the apical regions being more vulnerable than the other regions of the body, regions that are closer to the body. And this concludes that the visible damage is not equally distributed, and the results supported my hypothesis show that damage increases from the wing base onto the wing tip, and the apical regions experience the highest level of damage. For future studies, this could be include additional moth families and investigate how wind damage affects flight performance, as well as environmental factors such as rain, wind, and others, and then also comparing different habitats for species could be some of it. Something with my main takeaway from the study is that damage increases from the basal regions onto the apical regions, and understanding this wind pattern could give us an idea of how environmental and biological factors that affect moth performance as well as their survival and reproduction. And then my acknowledgements, I would like to thank Federico for helping me set up my light sheets, and then Leo for inspiring me for this project, as well as Frank for always guiding me. And then I'd like to thank the Bosque Eterno de los Niños field station for giving me the space to do this, as well as the Monterrey City. And then that is all for my presentation.
We have plenty time for questions.
Thanks, that was a really fascinating topic. I was wondering whether you considered alternatively that maybe the pattern of damage to the wings, rather than reflecting where they're most frequently damaged, could be reflecting survivorship bias. In other words, that the places you didn't find damage very often, it's not because they're infrequently damaged, but because if they're damaged, it's fatal to the moth.
Yeah. So, is that a question? Sorry.
Yeah, well, it's a suggestion, but you might have thoughts on that.
Yeah. So, that was one of my future resources, researches. Yeah. So you could also see what predators and the pattern as well.
Thanks.
Do you have any idea what the most common sources of damage are? What is actually causing different types of damage, or if you might identify that?
I don't know exactly what is causing these damages in these certain areas, but they are mainly through the hind wings from my experience. So I'm not exactly sure, but it could also be the main is probably predation.
What was the worst damage you saw?
Probably this one right here. Was probably one of my worst ones. I did have other ones that showed very, very bad damage, but I wasn't able to identify them. So that would be very interesting to see in future research.
Did you compare damage severity with the species or the size of the
I did not do size. I just did this specific moth family. Yes.
Do you think there is damage done simply by hitting obstacles as the moths are flying around?
Yes. And yeah, I do believe that.
No? Thank you. Thank you, Annie, for your help in this. Something to mention also, Lisa and people in this section of the symposium. Questions many times are very good source of points to add to your discussion or to improve some aspects, okay? So have that in mind. Next speaker, I don't know if he is here or is already outside the snake's body. Julieta from UC Santa Cruz.
Thank you. Good morning, everyone. My name is Julieta, and today I'm going to be talking about snakes in the dry season. So I did my study in Guajiniquil, which is in Guanacaste province, which is outlined on the right there. And it is dominated by dry forest, which is the picture on the right. So it's called dry forest because half of the year it doesn't get rain, and this makes everything really hot, and fresh water in general is very scarce. So organisms which live here have to adapt to it. The trees in this picture have lost their leaves during the dry season, and this is to help them retain their water. And so animals have to cope with these conditions in similar ways. In terms of snakes, it is a very diverse area. you can expect more than 30 species around the town itself. But this is only possible because not all of the snakes are active at the same time. So this big picture is a sea bond, and they eat snails. So during the dry season, there aren't any snails, and thus there's no reason for them to really be active. What snakes will do is estivate, which is essentially hibernation. They will go somewhere hidden away, and they'll wait for the rains in this case. At the very end of the dry season, there is the least favorable conditions for most snakes and animals in general. And this led me to my question, which snake species are active during an extended dry season? To answer this question, each night I would hike roads and trails. On this map, I colored in black the areas which I walked, and I've broken it into 3 main areas. There's Ukial, which is the more forested area on the top side. There's Escondida and Corona, which are 2 playas, sort of by this peninsula. So I grouped those as one area, including the roads that connect to it. And then finally, Murcielago is this last area in the bottom right. In total, I walked around 65 hours personally. Here are those hours in a graph. The graph starts at midnight, and then, like I said before, most of my hiking was right after dark. So where most of these bars is like around 7, and each of the black lines is a time which I found a snake. So in total, I found 16 snakes, 14 individuals, and 6 species. These are the 6 species I found. One of them in particular was much more abundant than the others. It's Trimorphodon quadruplex. I found them in all different sizes. Here's a picture of one that was 5 or 6 feet, but most of them were around 1, and some in the middle. Here's a map of where I found the snakes. I found 2 species in each of the 3 locations I mentioned, and none of the species overlapped. You can notice there's a higher concentration of snakes in that one area. Partially this is because I spent the most time walking there. So this graph, the x-axis is hours accumulated, and y-axis is total snakes found. So each time that the line moves up, I've found another snake. And one thing that is important about this is, while I mostly looked at night, I only found snakes during the night. And even when I looked during the day, I didn't find any snakes. So this is the one area where I found the most snakes. And the line is only increasing at night. So where you see these big plateaus, that's when I was looking during the day. And if I had looked during the night instead, I would assume that I would find snakes. Here's a closer look at that one area. I noticed some trends for where I was finding these snakes. So I found no snakes in the road itself. All of the snakes were either to the right margin nearby it, or some ways off to the left here. And this is for one main reason, I would say. Both of these areas shared steep rock outcrops. So these are good for snakes because snakes are cold-blooded, which means they regulate their temperatures through the environment. Usually snakes like to bask somewhere open because it gives them the most energy, but this also brings them dangers like predation. So basking from a refuge like these rocks here is usually colder, but in this dry season the temperatures are really hot, so they can bask here, get enough energy, and also be safe. The species which I observed the most, Trimorphodon, is specifically suited for these areas because they're good at climbing and they like to hunt in these habitats. Roads are not worth the risk, so they can get enough heat without using them, even though snakes would usually like to bask on these roads. This is a picture of that road where I found the most snakes, and you can see here the right side is much steeper than the left side, and it had more of that habitat that snakes are common in, which explains why they were on the one side. In terms of possible prey, lizards were by far the most common species which I encountered. There were many species of them of all sizes, so most of the small snakes would be eating these lizards on the left, but even the largest snake that I found could be eating juveniles of this iguana on the top. This picture is a snake which I recaptured. So I found it on one night and then again on the following night. And in that time which I had left it alone, it had found a meal. To me, I thought it was lizard shaped, but I don't know for sure. Lastly, here's a comparison of two species. On the right is Trimorphodon, which I've been talking about, and then on the left is Porthidium ophriomegas. Both of these species like to eat lizards, which they have in common, but what they differ in is the times in which they're giving birth or hatching. So, vipers on the left give live birth, and lyre snakes, Trimorphodon, hatch from eggs. But Trimorphodon hatched during the dry season, when I was surveying, and Porthidium hatched during the rainy season. So this could be because of selective pressures where they're dividing when they are hatching, and in this case, they don't have to compete with each other for the same reasons. So in summary, I found 6 species, mostly Trimorphodon, and I think this is because they like steep rocky areas, which provide heat and safety during the dry season, and their prey are very plentiful.
We have time for questions.
How did you identify the snakes that you recaptured as a recaptured snake and not another individual that's a snake?
So lyre snakes are very variable in their pattern. So by taking pictures of them, I could compare it, and mostly by like dorsally and their markings on their head are very distinctive. Right?
So obviously there's a lot of at least sweat. I don't know about the blood and tears, but can you think of another way, or do you know of other ways that folks do inventories or censuses of snakes? Or is it you hit the trail and you walk for hours and hours and you encounter them, and that's the way you do it? Could you do it with drones? Could you do it with cameras? Could you do it with dogs? There's gotta be people in the field looking for snakes.
I think it mostly is people in the field, but in terms of the actual methods, there are a lot of ways. In more temperate zones, during times when you're flipping cover, is very productive. But because the season was so dry, it's not as useful in an area like this.
Okay. Muchas gracias, Humberto. Okay, next speaker is Anna Drew from UC Berkeley, and she will come to speak about age and sex-related differences in food capture success by Regata Manduca.
Good morning, everyone. As Fede said, my name is Anna, and I will be talking about the Frigata magnificens, or the magnificent frigate bird. And this picture here is of a juvenile. Magnificent frigate birds are large tropical seabirds found throughout the tropical Atlantic and eastern Pacific, including areas along the Pacific coast of Costa Rica. They are highly specialized aerial foragers, meaning they obtain nearly all of their food while flying. Unlike most seabirds, they cannot rest on the ocean surface because their feathers aren't waterproof and would become waterlogged. Instead, they capture fish directly from the water surface or steal food from other birds through kleptoparasitism. Because food acquisition occurs almost entirely birds provide an excellent system for studying competition and feeding success. So, how do I identify them? Males have all black plumage and a red golar sac, which they inflate during mating, like was showed on the past slide. Females are significantly larger. They can actually be up to 25% heavier than males, making them one of the few seabirds where females are larger in size. And then juveniles always have a white head, which is very distinct, and a white chest. These differences are important because body size, flight performance, and experience may influence how successfully birds compete for food, and this observation led directly to my research question. So, because males, females, and juveniles differ in both morphology and experience, I wanted to know whether these differences translate into differences in feeding success. So my question was, are there age and sex-related differences in foraging success in frigate birds when presented with food? My predictions were that adults would outperform juveniles because of greater flight experience and competitive ability. Specifically, I expected males to have the highest success, females intermediate success, and juveniles the lowest because of preliminary observations. So I conducted my study in Picote Bay in the Gulf of Santa Elena in Costa Rica. I had 5 days where I conducted 10 feeding trials, and trials were conducted from the back of a boat in open water. For feeding trials, I used 10 mackerel fish cut into 40 pieces of bait, and 393 pieces were thrown in total. Fish pieces tossed individually into the air, just at random, and each feeding session was 7 minutes long, and there were 20-minute intervals between trials. This is a little video clip of me conducting my experiment. Again, I just throw the fish at random and record video to see who captures it. All feeding interactions were recorded, and individuals were better identified through pausing and going through the video. And here's a video example of a male successful catch, a male unsuccessful catch, and the male gets it. The male misses. And then the juvenile misses as well. So my first graph shows the total number of successful captures recorded for each age and sex class. The horizontal axis shows the 3 groups, males, females, and juveniles, and the vertical axis shows the number of fish pieces successfully captured. A total of 206 successful captures were recorded. Adults accounted for 100, adult males for 137, females for 38, and juvenile for 30. Now this graph shows the percentage of successful captures contributed by each group. Males accounted for about 66.5%, sorry, wrong graph. There were 325 males present throughout all 10 trials. They caught 137 pieces of fish, and they had a success rate of 42.2%. Females, there were 78. They caught 38 pieces and 48.7%. And juveniles, there were 149 throughout the 10 trials. They caught 30, and they had a catch rate of 20%. The most important finding from this study is that food capture success differed substantially among age and sex classes. Juveniles were much less successful than adults, suggesting that experience and aerial skill play an important role in food acquisition. Interestingly, females show the highest capture efficiency despite males capturing the greatest total number of fish. And these findings are consistent with UC EAP student Brandon Hammergren from fall 2023, and she found that adult frigate birds were more successful in obtaining food and played dominant roles in feeding interactions. Together, these studies suggest that age, experience, and competitive ability strongly influence foraging success in magnificent frigates. This matters because feeding success directly affects energy intake, survival, and fitness of frigate birds. It helps us understand patterns that help explain behavioral interactions and population dynamics within seabirds and specifically frigate birds. And the results contribute to our understanding of how age and experience shape ecological performance. In future research, I would definitely increase sample size, collect data across multiple seasons, use individually marked birds to track repeated behavior, examine how body size and flight performance affect feeding success. And these are my acknowledgements. I learned how to drive a boat, so that was a lot of fun. And thank you to my advisors, Frank and Heather, for that.
We open the period for questions.
Yes.
Do you know why there were so many more males observed in your study?
I do not, as breeding season is not really known in Costa Rica. I would assume that would be why, but I'm not sure. Yes.
Could you talk about how throwing a piece of fish kind of indicates like the natural thing? Is that, or did you look at any other methods to kind of indicate like how comparisons?
I did look at trying to get the fish to float. Getting them to float was a little bit harder than I expected, so I did just go with throwing them. But if I wanted to conduct more naturally accurate research, definitely floating fish would be the way to go.
Yeah.
I have a more general question regarding the differences between shape and size of males and females. Do you have, if there is an explanatory answer regarding that?
Not that I know of.
And associated to this question, do you know if there are differences in the parental care that they exhibit?
I know it's dual, both mom and or male and female provide parental care, but that's
And distance of life, something like that? Thank you. I was just curious.
And like when you are throwing and doing this observation, and you have maneuverability, ability, and speed, what do you think these birds are better doing?
like when I throw the
If they are faster or they are maneuvering? The successful is related with one of these or
I would think the maneuver would go.
that's it. Thank you, Anna. If you are doing, Tanya, I should learn how to do it. Keep on going with animals with feathers. Next speaker. Gracias, Jaime. Let me introduce next speaker, Caleb Mitchell from UC Santa Cruz. He's going to speak about mixed flocks composition along Monteverde from urban to rural breeding.
Alright, hello everybody, thanks so much for being here. My name is Caleb Mitchell, and today I'll be talking about the avian mixed species flocking behavior and species composition along the urban rural breeding. Okay, so for a little bit of background, many species of animal tend to exist in mixed species groups. These are groups that are defined by inter-specific interactions. Animals tend to join these groups for a variety of benefits, usually a boost of fitness either through increased health and safety or increased foraging opportunities. one of the most notable examples of mixed species groups is in birds, especially songbirds in the family Seridae. They are they're highly social and will form large flocks composed of many different species. There are two main species, or two of the largest reasons for flocking songbirds include predatory avoidance and foraging efficiency. Essentially, the benefits they get from a bunch of functional purposes within their programs, or within their flocks include the many eyes effect, basically guaranteeing increased safety for predators, knowing that there's always a pair of eyes watching, allowing them to forage more often, and the dilution effect, meaning that any given individual is much less likely to be the one targeted by a predator. Flocking behavior in general is seasonal in many regions, especially the more temperate areas of the world where there is a much clearer split between the breeding season and off-season, where during the breeding season, usually during the springtime, individuals will pair off and exist in breeding pairs rather than flocking. However, here in the neotropics, mixed flocking, especially mixed flocking, persists as species will join mixed flocks in breeding pairs. So here in the neotropics, these flocks tend to compose of nuclear species and attendant species. Nuclear species tend to be much more loud and conspicuous, very easy to follow, while attendant species, again, tend to be those pairs that love to join for the added benefits. However, despite all of the benefits, urbanization presents a significant disturbance to social behavior within birds. Previous studies in the temperate regions of the world have shown a negative correlation between constructed area and species richness within songbirds. However, it has yet to be shown exactly how this affects these more tropical flocks persisting into the breeding season, which led me to my central question, examining the effect of urban development in Monteverde, Costa Rica, on the size and composition of tropical flocks during this time period. As for my materials and methods, I surveyed 4 different reserves within Monteverde, or 3 different reserves, 4 sites. One of my sites was downtown Santa Elena, much more developed than the rest of Monteverde, and served as my urban core. While I also surveyed the Santuario de Monteverde, Bosque Eterno de los Niños bajo Tigre, y the Corricancho Reserve. Each of these is situated in a rough line of about 3.4 kilometers between Santa Elena and the Corricancho Reserve, creating a gradient of sites, increasing distance from the urban core. As for data collection, I visited each site between 7 o'clock and 11 o'clock a.m. Between the 13th and 23rd of May. I surveyed each reserve twice, walking slowly along the trail, listening for nuclear species to find flocks, finding them as 2 or more birds of different species existing within 20 meters of each other for 3 minutes or more. As for my results, overall species richness, we saw just a flat, nearly linear increase the farther you got away from the urban core. In total, I surveyed a total of 16 flocks across all 4 sites, composed of 99 individuals of 17 different species. 27 different species. Flocks found were also very similar to the species richness, just seeing a general increase the further away you got from the urban core. Average flock size was a little more interesting, but still saw greater numbers further away from the middle of town. And here I have a 4-way Venn diagram comparing the overall, or the relative species richness and overlap between sites. as you can see, there was much more of the species than not were exclusive to their sites. There wasn't a ton of overlap between sites, which I will talk about. That's for my discussion. The species richness and number of flocks all increased with distance from the city. This was expected. It lines up very well with past research in terms of flocking behavior in temperate regions. It just suggests a disturbance in flocking activity due to urban activity. My species composition did very heavily between sites, which was a little more unexpected. I expected quite a bit more overlap. However, this could be explained by just a relatively small sample, as well as the fact that urban cores worldwide tend to be a bit more homogeneous. Many are heavily exploited by certain species, usually crows, blackbirds, and doves, which I saw quite a bit of in the urban core, which weren't counted since they weren't participating in any mixed flock behavior. I also encountered many breeding pairs, both within and independent of flocks, supporting this idea that they like to join these flock within family units. However, not many were included within, or I wasn't able to take comprehensive data on the number of species within these flocks that were participating in mating behavior. This could have, or a future study on this could have a profound effect on the conservation of birds in Monteverde, especially as protecting reproductive habits of songbirds or anything is very vital to conservation efforts in terms of urbanization, especially as Monteverde's ecotourism industry continues to grow. Acknowledgements I would like to thank them, Puerto Rico, my advisors, as well as Leo and Frank for offering general advice in the program set on a certain path, as well as each of the reserve directors for allowing me to serve in the grounds.
Okay, it is time for questions. About 3 minutes. I will go ahead.
So, exactly where in Santa Elena did you start your look for mixed species flocks? And just to say, Santa Elena, maybe next to San Jose, would be the last place that I would look for a mixed species flock. But it's very interesting. Yeah, cool project.
Yes, yeah, that was kind of precisely why I wanted to survey the city, because I wanted to see I did expect there to be less activity within the urban core, but I did just want to establish that gradient and kind of examine the extent to which how much flock behavior was disturbed by urban activity. I did start my observations. It was kind of a wide area within town, but it a lot of my observations were near or between Banco, Costa Rica, and the very center of the markets in Santa Elena.
Pardon me if you said something about this and I missed it, but did you anecdotally notice any differences between what the birds were doing in the different habitat types while they were in the flock?
A bit. I mean, there was a lot of anecdotal things that I noticed, especially I saw quite a few breeding pairs, especially I do believe I saw more in Coricancha, which was the farthest side from the urban core. Other than that, though, it was a little difficult to define behavior within the dense urban cores.
Okay. Looking the report I saw in the list, a couple of species that I don't know how did you consider participating in one of these groups. One is the motor and the other was the toucan. So those, how, which was the criteria to include species like that?
Right. Those were a bit surprising to me too. I just included them as so my criteria was whether they, or just any two or more species existing roughly 20 meters of each other for more than 3 minutes. I only found, or I did find, both of those were those individuals that just seemed to be moving a bit with the song group passing through, so they could get more as part of the songs.
I could imagine them trying to eat somebody. You have more questions? No? Muchas gracias, Kayla. I never can. Gracias, Tania. So finally, we will move from feathers and scales to hairs and listening about projects related to mammals. So first is Kaylee Vandermark from UC Davis. Is going to speak about bacterial and fungal biodiversity in sloth fur.
Okay, this is my project on the bacterial and fungal diversity in captive sloths. For my project, I focused specifically on the species Choloepus hoffmanni, Hoffman's two-toed sloth. I chose to focus on sloths because they are often called mobile ecosystems due to their fur containing multi-trophic communities that interact with both abiotic factors such as the temperature and the weather, and biotic factors such as the organisms living in their fur, which include various types of algae, bacteria, fungi, and arthropods. Specifically moths are common in their fur. They also have a unique fur structure with various cracks and grooves in them that are speculated to help facilitate algae growth as well as the growth of possibly other organisms. This specifically focuses on fungi and bacteria. There was not a lot of information on the types of bacteria that can be found in sloth fur, but for fungi, nearly all fungi that are found in sloth fur are ascomycetes, and most of them belong to the order Capnovialis, of which the family Neodeversiaceae is the most common. The picture you see here is the Silvatura Park sloth habitat. This is where the sloths I got fur from were living. My central questions for this study were how many types of fungi and bacteria are generally found on one sloth? And is there any bacteria or fungi that are particularly common within sloth fur? I obtained fur samples from 4 adult female sloths, and from there, I cultured them after creating 20 petri dishes. These petri dishes were sterilized via autoclaving, and I used 2 different types of agar. One was potato dextrose agar for growing fungi. And the other was lower Hinton agar for growing bacteria. I placed about 0.01 grams of fur in each petri dish and placed the PEA dishes into a drying rack that maintained a temperature of about 25 to 29 degrees Celsius throughout the day, and the MHA placed in an incubator that was set for 38 degrees Celsius. I checked on them about once per day after that until they had sufficient growth, and then viewed them under a dissecting microscope, where I separated them into different morphospecies based on shape, texture, and color of the growth. I found a total of 14 different bacterial morphospecies and 8 fungal morphospecies. The first sloth that I looked at had 3 fungi and 5 bacteria in it. The second one had 1 fungus and 4 bacteria. The third had 6 fungi and 5 bacteria. And the fourth had 3 fungi and 5 bacteria. I made Venn diagrams to show the overlap between each sloth as well as which ones are unique. You can see most bacteria were unique to each sloth. There's 3 that overlapped between them. See the bacteria M overlapped between just sloths 3 and 4. Bacteria B overlapped between sloths 1, 2, and 3, and F between sloths 2, 3, and 4. Most of the fungus found was on sloth 3, which had double the amount of fungi compared to the sloths with the second most fungus present. Most of this fungus was unique to that specific sloth, but it also overlapped. It had a fungus A that it shared with sloth 1 and 4, a fungus F that it shared with sloth 4, and the other one that overlapped was between sloths 1, 2, and 4. These are the NHA plates for growing bacteria. You can see that sloth 2 had the least amount of growth out of all of them. And these are the PDA plates for the fungus. You can see that sloth 2 also had very little growth in this one, and sloth 3 had a much greater amount. The number of bacterial morphospecies per sloth had pretty low variation between bacterial morphospecies compared to the fungal morphospecies. And there was also no bacterial or fungal morphospecies that were found across all 4 sloths, which was rather odd because 2 of the sloths were all over each other. And so they shared like nothing between what was found in their fur. I thought there would be more in common with those 2. Previous studies have shown a lot more fungal variation between sloths. A study by Higginbottom in 2014 showed 84 different types of fungi across 9 sloths. And a study by Rojas-Batchins in 2023 showed 3,164 amplicone sequence variants across 20 fur samples. Possible differences between these studies is that they both included a different type of sloth, the Bradicus variegatus, which is a 3-toed sloth, which is known to have higher fungal diversity than 2-toed sloths. And it is also important to note that wild sloths likely have different fur composition. The results from wild sloths would likely differ a lot more due to environmental differences, such as the openness of their habitat, the weather, such as rain or wind, and interactions with other animals. I would like to thank my primary and secondary advisors, Naomi and Federico, the veterinarian that allowed me to collect fur samples, Janet Sandy, Maricruz Hernandez, who actually collected the samples, Silvia Park, for allowing me to use their sloths, and Monteverde Institute for allowing me to use their lab. Thank you.
Open the period for questions, please.
Do you know why they have so much algae and fungus on them?
They're rather slow moving, just most of it.
And do you know if it like helps or harms them?
Some some people speculate that they eat the stuff that grows in their fur, specifically the algae, but there's no actual proof of that.
I have a question, maybe I just missed it, but how you control the growth of fungi versus bacteria in those plates?
I use different agar to help with that, but I think there was bacterial growth in the fungi plates. It looked different in texture, so that helped separate it. But there was no fungal growth in the bacterial plates.
And the other one, can you link these differences in microorganisms' presence to some specific condition for each slug?
Not that I know of, because their enclosure was indoors, so they were rather protected from the outside environment. These two that I was talking about were the only ones where I might have looked into it, but they didn't have much of anything in common. These were slots 1 and 4, I think. They did have the same amount of bacteria and fungi, just none of them were not.
Do you have any sense of, like in other mammals, what the microbial fauna and flora might be? Whatever it happens, yeah.
I don't know about for fungi and bacteria, but there's 2 other mammals that are known to have epibiontic algal growth in their fur, and those are captive polar bears specifically, and I think it was manatees.
You can listen that like grooming and cleaning themselves behavior is important, but how this is happening in slugs? Do they groom themselves or show them? So they carry what they carry. It's very interesting that being in the same place in contact, they have very different types of things growing. If there is any other question, we can say thank you, maybe.
I love pork.
So, thank you again. Now, we're going to listen to the most interesting presentation of the symposium by Alexander from UC San Diego. He's going to speak about antibiotic resistance in oral microbiome from common vampire bats. Thank you.
All right, good morning, everyone, and thank you for being here. As Fede already stated, I'm going to be just discussing a little bit about the antibiotic resistance in the bacterial microbiome of common vampire bats, as well as comparing that to the microbiome and antibiotic resistance in other species as well. Okay, so for a little bit of introduction, bats have been highly studied recently due to the increase in zoonotic disease transmission. Especially common vampire bats have been studied heavily in regards to rabies transmission and prevention protocol. However, the bacteria in the mouths of common vampire bats has not really been studied, which I find kind of interesting because the way that they eat is they make a small cut in the skin of an animal and they lap up the blood that comes out. This can introduce bacteria from the mouths into the open wound of animals, which is very important for us to monitor, especially since they're, the animals that they prefer to eat from are cattle. And then, as far as antibiotic resistance goes, it's now becoming a huge global problem. So it's important to start monitoring antibiotic resistance in many different bacterial colonies around the world. Alrighty, so I got a couple of central questions here. The first is, what bacteria is present in the mouths of common vampire bats and how it differs from other bats? And then how does the bacteria in the mouths of these bats respond to common use antibiotics for cattle? All right, so to start off the methods, we did use mist nets on the Gwendon farm to catch as many bats as possible. Both common vampire and non-vampire bats were untangled, identified, swabbed using sterile swabs, and then re-released into the environment. We used 2 sterile swabs for each bat. One was used for an agar plate to run the antibiotic resistance test, and the other one was used for a separate plate to monitor the diversity. All right, the filter paper disc diffusion method was used to test the antibiotic resistance of the bacterial colonies present in the saliva of these bats. So basically what that is there are 2 antibiotics that I used and a control, which was distilled water. The 2 antibiotics I used were oxytetracycline and gentamicin, both brought down to the concentrations that you see here. And the filter paper discs were dipped in these solutions and placed on an already striped agar plate. They were then stored in the incubator at 38 degrees for 72 hours. The reason that they were stored at 38 degrees Celsius is because this is the internal temperature of a cow. So I was trying to kind of replicate the wound site that they would naturally go on. The plates were then removed and photographed for processing using ImageJ software. And then the different morphospecies present on each plate were identified under a dissecting stripe. The morphospecies were identified and labeled using 7 different physical traits. Colonies with the same physical traits as others were considered to be the same morphospecies. For each plate, the number of colonies of each morphospecies present and what morphospecies they were was counted and recorded, even for the plates with antibiotic treatment. ImageJ was then used to measure the distance from the discs to the nearest bacterial colony. This was used as a proxy for measuring antibiotic resistance patterns. So essentially, the ones that had more space from the filter discs to the colonies had lower levels of antibiotic resistance associated with the antibiotic, and the ones that had bacterial colonies touching or growing over the discs were considered to have high levels of antibiotic resistance associated with these antibiotics. A student's t-test was used to compare the richness of the morphospecies found in the plates with antibiotics and the plates without antibiotics to see if there was a difference in the diversity of these plates. And then a Crystal-Wallace test was performed to identify any significant differences in the antibiotic resistance between treatment groups. All right, so for the results, we caught 20 bats altogether in the mist nets. One of the bats, unfortunately, could not be identified and was not swabbed. One bat was actually recaught on a different day and was swabbed, but we did not use the data for analysis. All right, so for diversity, this is actually really interesting. We didn't find a significant difference in the richness between the samples that were from the common vampire bats and the ones that were from the other species of bats. Up here on the top, we have kind of a breakdown of the abundance of each of the different morphospecies. I know it's a little bit small, but basically all of these little points here are just the morphospecies tags, which wouldn't really make sense without the diagram to explain them. But essentially what it's showing is it's showing that most of the morphospecies abundance was kind of dominated by like 3 to 5 different morphospecies for each of these sample groups. Okay, for another look at the diversity between the common vampire bat morphospecies and the non-vampire bats, we have graphs that contain essentially all of the morphospecies that were seen in 2 or more plates for each group. And what's really interesting actually is that for both of these graphs, the first 4 from the left are seen in both groups, which means that they have very similar bacterial colony diversity. So it doesn't actually vary a lot in the richness, and it doesn't vary a lot in the, like, what the morphospecies are between these two groups. All right, this is another look at diversity. However, this is a look at the diversity and the richness between the plates with antibiotics and the plates without antibiotics, broken down by common vampire bats and non-vampire bats. As you can see here, there's not really a huge difference in the richness level between the different plates. So this means essentially that even though there's antibiotics present on one of the plates, this really didn't impact the richness levels that we saw. All right, for antibiotic resistance, there was high levels of antibiotic resistance that we saw. The average distances from each of the recorded treatments was measured here, and then there's the standard deviation next to it. As you can see, the standard deviation is a lot larger than the average distance. A test was run to see how different they were, and there was no significant difference found between treatments, which includes significant differences between the antibiotics and the control. Discussion. Okay, so there's not a huge difference in the bacterial morphospecies richness between common vampire bats and non-vampire bats, as well as the fact that there was a huge overlap in the morphospecies identified for both groups. This means that the bacterial microbiome of both groups was very similar. There was also not a huge difference in the treatment effectiveness for preventing bacterial growth, which essentially suggests that there is a high level of antibiotic resistance in the bacterial colonies found in both common vampire bats and non-vampire bats. Because there is no significant differences noted, this does unfortunately mean that the antibiotics were ineffective in preventing bacterial growth. All right, some things to consider is that the method of measuring antibiotic resistance could have erased small effects that the antibiotics had in the colonies. So for example, as you can see here, the way that we measured it was just that if it was touching, if there was a bacterial colony touching these discs, it was measured at 0 millimeters distance, which is the highest level of antibiotic resistance that these treatments can get. However, there is a difference in the way that these colonies are responding to the antibiotics. As you can see here, one colony of the same morphospecies is touching this disc here, but this one that's the same morphospecies of a different colony has steady growth all around the ring, except for when it gets to the gentamicin treatment group here. It shows reduced growth, and then it continues going. So that's an important thing to consider. And then another thing was that antibiotic resistance for each treatment group was not separated by morphospecies. So for further research, I would recommend separating this by morphospecies to see if certain morphospecies showed a preference or resistance to different antibiotics. Thank you to everyone who helped me with this. Fede, my primary advisor, who helped me with every step of the way. Naomi helped me in the lab and was my secondary advisor. Minor, thank you so much for all of the coffee and support. Bendito, Wingon for helping us, showing us where the cows were every day and letting us use the farm. Thank you to everyone at MVI who helped me here get into the lab and use that, even when it was super late at night. And thank you to all of the veterinarians who helped me procure the antibiotics. Are there any questions?
You said you incubated the plates at a certain temperature to kind of imitate a cattle temperature. Did you ever consider the fact that this temperature could vary within cattle in different regions or within cattle at different times of the day, etc.?
Yes, so I did consider that, as well as the fact that the temperature on the skin may be different than the true internal temperature. However, I'm not able to really replicate that in this lab setting, especially since other people were using the incubator and we needed it at a steady temperature for a longer period of time.
How did you determine the concentration of the antibiotics you were using? Because naturally, if you use the higher concentration, there would be less release.
Yeah, so the concentrations were essentially the lowest effective level as seen in other clinical studies, and the lowest effective level is usually what is used for antibiotic resistance tests just in the industry.
I'm just curious, again, maybe I just missed it, but which kind of antibiotics did you use?
I used oxytetracycline, gentamicin, and the control was distilled water, which was used to make the solution.
And why you choose that?
The antibiotics I chose were used very commonly in cattle, especially as topical antibiotics. So they were the most kind of relevant for the problem that I was looking at, not to mention that they were the ones that I was able to procure.
And do you think that maybe ivermectin has a different effect in this?
Yeah, so ivermectin is actually a dewormer and it would definitely have a different effect, but I'm not sure how it would help as an antibiotic.
Okay, and the other thing, just I'm curious, did you use a extraction chamber when you were incubating or using these bacteria?
I'm sorry, what was that?
Did you use an extraction chamber while you were working with bacteria?
I'm not familiar with that.
We don't have them. Megan?
So, how would you suggest moving forward then? What do you think is the application, knowing that these aren't very effective antibiotics?
Yeah, so that's definitely very concerning. We should definitely do further research. Like I said, there is definitely some response to the antibiotics, but to what level we were unable to determine during the course of this project. So further research, separating the most common morphospecies to their own place, and then looking at maybe like a breakdown of how they respond to the antibiotics over time would be very interesting to see if they have just a natural antibiotic resistance or if this is something that they develop as they grow.
Did you mention how did you avoid fungi growing in your place?
I did not, but to avoid fungal growth in my plates, I made the agar plates and mixed in niacin, which is an antifungal agent, and this allowed me to be sure that the organisms I was looking at were bacteria.
In this presentation, we have finished the second section of the symposium. We're going to have another break and another amount of food. I don't think you are able to eat all of that. You can try. Our guests have the priority in going to the table. Thank you. And before Richmond, let's see some people going in 10 minutes.
Yeah, there's like a bit of
Thank you.
yeah, that's, I'm on I've kind of been just
it was right behind a garden. So the garden plants would come over, but I tried to make sure I only picked the ones that were growing on the gardens.
When they grow in rocks and metal, they are very good. Do you, I know that your results of the soil is present actually, but I am curious about how did you evaluate, how did you use your visual, or did you prove or something the soil? I am curious about how there is variation in depth or in how much it is
Yeah, all of my soil observations visual, and whether it was at the site, and then I checked it again because I took a look at the plant, so I checked it again once I was analyzing my data to make sure that it matched with what I had read about the site.
My experience with plants growing in non-conventional substrates is that other people do the best that they can to get rid of them because they cause structural damage. And gotten into some fairly animated discussions, to not say fights, because in some cases, if you leave a plant growing in concrete, it finds its way down and splits the concrete. So I'm very, very intrigued by the notion of like letting plants in urban areas just grow and do their thing. But you might be like swimming against the current to try to let that happen. How would you yourself balance between the destruction that plants do, and some of them, not very teeny ones, but bigger ones, or ones that start small and get bigger?
I think it needs at least plants in urban spaces. I think it needs to be planned and designed. So having designated structures that are built for plants to grow on is really important, and knowing what plants are growing on them and making sure that they won't damage the built environment is also really important.
Awesome answer. I've seen a couple places where there is a wall of plants that all
Somebody should give Yellow a mirror.
This is going to be quick because taxes are the key here soon. I'm picking up to the station to eat lunch and stuff. But as far as closing remarks go, I'd just like to say a couple of fast things. One, an enormous thanks to Joyce for translating. For the people who don't speak English, it's really an awesome opportunity to learn what you folks did. Also, tremendous thanks to all kinds of people and institutions, both Northwood and Institute, resource people, and especially other instructors, as in Federico, Leo, Naomi, and other people who helped make this happen. And lots of people way outside what I can say right now. There will be other times actually Saturday night, there'll be another opportunity to thank and recognize people. But I would also like to point out that it was 70 days ago today, so today's Wednesday, where we met in the Hotel Pax on the 25th of March. And it's always really a little intimidating, a little exciting and fun to hear what you have to say and what your expectations are about doing research and doing projects. And we want to do everything, we want to do it really well. And the script is not written, it's like the outcome of, an athletic event. We have no idea what you're going to do and how you're going to do. But everyone did really an excellent job. And one of the things that struck me in this symposium presentation was that everyone excels at something. Maybe there were a few things that could use improvement. Gosh, you wouldn't want to do an absolutely perfect project or perfect presentation because you want to keep going, perhaps. Maybe you don't. Maybe you say, hey, I've had enough of doing research in the tropics and I'm going to look for an air-conditioned space, and that's my fate. That's what I want to do. But at least now, you know what it's like to come up with a project on your own, to struggle through the process of making it happen, collecting data, and then trying to make sense of it. And so I would like to sincerely thank you for your effort and for getting here and going through the process. It's a tremendous sense of fulfillment and satisfaction and relief that we get to this day. And the presentations today were really, really meaningful. Thank you. Just so you know, instructors are going to go up to the station at around 2 o'clock or so you will be there, but I'm not expecting you to be cranking on anything other than recharging your batteries however you wish to do that. Go to town, go shopping, take a nap, go for a walk, go running, eat, whatever you want to do. It would be probably a smart idea to sort of collect things, maybe do a little pre-packing or whatever, know where your passports are. Maybe could you just look around? I'm going to close my eyes and ask. Does everyone know where their passport is? But I don't want to know yet. So you did a great count. Okay, so don't count other people. Don't count. Just
I know where is my passport.
You know where your passport is? Okay,
I don't care.
But you're not. Okay. Do you know where your passport is? We good?
We are totally good.
Oh, good. Awesome. And do you know where yours? Just say, I do, yeah. Just in case the policía de migración comes at the customer.
You know, I don't have my cedula. I don't have my phone. I was in a rush this morning.
Oh, and what's the fate of your phone? Has it really died?
No, thanks to Haley, it is resuscitated.
Wow.
Yeah.
All right. So see you up at the station in a little bit. Again, thank you. Awesome job.